There are more possible drug-like molecules than there are stars in the universe, around 10 to the 60th. Somewhere in that ocean are the few that could become a medicine. So how does anyone actually find them? In this series, we're not just going to talk about it, we're going to do it, in code. By the end, you'll run your own virtual screen against a real drug target. Let's start at the beginning. Drug discovery is a search problem, and the strategy is a funnel. You start with a huge library of candidate molecules, maybe millions. Then you narrow, stage by stage. First, keep only the ones that look like drugs. Then, the ones chemically similar to molecules already known to hit your target. Then, the ones a model predicts are active. Then, the ones that physically dock into the target's binding site. Each stage is cheaper than making a molecule in the lab, so you spend computation to avoid spending money, and reserve the slow lab work for the tiny handful that survive to the bottom. Two things anchor everything: a target, usually a protein driving a disease, and, if you're lucky, a set of known active molecules that already hit it. That gives you two ways to search. Ligand-based, learn what the known actives have in common, and find more like them. You don't even need the protein structure. Structure-based, take the protein's 3D shape and test how molecules fit into its binding pocket, by docking. We'll do both. Our running target will be EGFR, a kinase behind many real cancer drugs. Here's the good news: the core tools are free, open source, and it's basically all Python. The star is RDKit, the Cheminformatics toolkit that lets you load, draw, and analyze molecules in code. We'll pull data from ChEMBL, a public database of bioactive molecules. We'll use scikit-learn for machine learning, and smina for docking later on. And we'll work in Jupyter notebooks, so you can run every line yourself. Which brings me to the GitHub repo. Every episode is a notebook you can download and run along with me, the link's in the description. Clone it, create the environment with one command, and you're ready. This is a code-along, pause, type, experiment. That's how it sticks. So let's write our first real code. We import RDKit and load a molecule from a SMILE string, a compact text way to write any molecule. We'll use aspirin. That one line of text is a complete description of the molecule. And when we ask RDKit to draw it, there it is, the actual chemical structure of aspirin, parsed straight from text. That's the core magic: molecules become objects you can compute on. Now the useful part. Molecules have properties that predict whether they can be drugs, and RDKit computes them in one line each. Molecular weight, how big it is. Log P, how greasy versus water-loving, which affects absorption. And hydrogen bond donors and acceptors, which affect how it interacts with the body. For aspirin, a molecular weight around 180, a log P of 1.3, 1 donor, 3 acceptors. Small, balanced numbers, the profile of a clean oral drug. Those four numbers are the heart of the most famous rule of thumb in drug discovery, Lipinski's rule of 5. Oral drugs usually have a molecular weight under 500, a log P under 5, at most 5 hydrogen bond donors, and at most 10 acceptors. Let's turn that into a function and score a few molecules, including erlotinib, a real EGFR inhibitor, and reserpine. In a few lines, we get a table: aspirin, caffeine, and erlotinib all pass, but reserpine fails, on molecular weight. And there they are, side by side. Notice erlotinib, our target's real drug, sitting comfortably inside the rules, while reserpine, a larger natural product, breaks one. That's the rule of 5 doing its job as a filter. In episode 4, we'll run this exact logic across an entire library, to trim thousands of molecules down to the promising few. So that's the foundation. Drug discovery is a funnel. You start from a target and known actives, and the whole thing runs on free Python tools, with RDKit at the center. You just loaded a molecule and measured it in a handful of lines. Next episode, how computers really represent molecules. The notebook's in the repo, see you there.